Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from Bayer.
This program is presented by Medscape Education Global.
Hello everyone, and welcome to this program entitled Advancing Precision Cancer Medicine, Unveiling the Future Frontier. My name is Elena Garralda. I'm a clinical investigator, medical oncologist, and I'm director of the Phase 1 program and co-director of the clinical research program in Vall d'Hebron Hospital in Barcelona, Spain. In this activity, Atish Choudhury, Narushka Naidu, and Umberto Malapelle and myself, we will be discussing precision cancer therapies and techniques that show promise for the future. In this first chapter, I'm going to be discussing the next generation precision immunotherapy. So it is clear that immunotherapy and PD-1, PD-L1 inhibitors has changed the landscape of the treatment of cancers. However, the reality is that still multiple tumors won't benefit of PD-1, PD-L1 inhibitors, and there's still a lot of work to be done in order to improve the results in more patients. Some key challenges that need to be addressed is understanding the mechanisms of anti-tumor activity, understanding drug resistance, improving in the search of biomarkers, and finally finding rational combinations that enable patients to really benefit of immunotherapy. There are emerging technologies when we talked about precision immunotherapy. The first one is adoptive cell therapy. We're talking second one about vaccination, 3 T-cell antigen-directing antibodies, and finally, how can we improve immune checkpoint inhibitor blockade? So when we talk about adoptive T-cell therapy for solid cancers, we talk about different modalities of treatment. This is, remember, a living T-cell therapy where we're going to extract the T-cells from different sources. In the TILs, the source is the tumor, and the TILs are obtained from the tumor. And here, the T-cells will be infused and recognize the tumor's T-cell recognized by the MHC and the tumor antigen. TILs have been approved, and it's the first T-cell therapy that has been approved for solid tumors with the approval of lifileucel in February 2024 for melanoma patients. We also have engineered TCR T-cells, where the T-cells are obtained from the blood and they are transduced with an engineered TCR that will recognize the antigen bound to the MHC. So this has some specification of MHC recognition. And we also, in 2024, we've had an approval of an engineered TCR, and this is afami-cel for HA4 positive, HLA-02 positive synovial sarcoma. And finally, we have CAR T-cells, where the T-cells are actually also obtained from the blood, but they are transduced with a CAR directly, so not an engineered TCR, but a CAR construct that will recognize the antigen that is bound to the membrane. So we don't require any MHC recognition. So just as a brief reminder, TIL treatment is a one-time 3-step treatment that requires high-dose lymphodepleting chemotherapy, the intravenous infusion of the expanded TILs on day 7, and then high-dose interleukin-2. All patients will have grade 3 and 4 toxicities, and a lot of effort is being done to try to improve this treatment. Just to say that in these last years, we've seen huge successes. This is the only phase 3 trial that we have comparing TIL treatment versus ipilimumab in patients with melanoma. It's a positive trials for TILs. And here is the phase 2 trial that has led to the approval of lifileucel by the FDA in patients with refractory melanoma, response rate of 36% and a complete response rate of 3%. And some of these responses are durable. Work is being done to select better TILs, to improve the T-cell performance, to improve the tumor microenvironment, and to reduce treatment toxicities. Just as an example, in Vall d'Hebron, we are working, for example, to try to select the neoantigen reactive T-cells, as we believe this is probably the T-cells that are generating a bigger success. both in patients with melanoma and other epithelial tumors. And similarly, we are actually also running at a clinical trial where we compare TIL versus high-dose interleukin-2 with a modified interleukin-2 to try to reduce the toxicities of the high-dose interleukin-2. Regarding engineered TCR T cells, as I mentioned, we do have the approval of the MHA4 positive, HLA-02 positive, and this here you can see the phase 1 results with an overall response rate of 24% in all solid tumors and a 44% when we talk about synovial sarcoma. This led for a phase 2 trial that has led to the approval, but also other opportunities to be explored in other tumor types and different antigens that are being explored in different tumor types. Finally, CAR T cells, the reality is the efficacy of CAR T cells in solid tumors has been limited so far, and this is mainly due to the lack of highly cancer-specific antigens, which create significant toxicities of tumor on target, and also of the poor expansion and scarce persistence of CAR T engraftment and issues with the tissue distribution and tumor penetrance. Even so, we're starting to see some advances in the field, and here you can see, for example, an example of a CAR T targeting Claudin 18.2 in GI tumors with an overall response rate of 57% and a disease control rate of 75%. Finally, just to mention that there are multiple targets that are being studied for the cell therapies, such as mesothelin, HER2, B7H2, and different hematological ones. And just to mention that on top of CAR T cell therapies, we have other cell modalities, such as NK cells, that are being also explored. The second modality of treatment would be the bispecifics, and here we have engineered artificial antibodies capable of recognizing two antigens, and you can see the different formats in the slides, really different variety of treatments, and the rationale is that you can have a greater potency and selectivity compared with a combination of two monoclonal antibodies. A specific type of would be the T cell redirection in bispecific, where we bring the T cell into the tumor to actually create this killing of the cell tumor. Bispecifics in immunotherapy are being tested in different ways. As I mentioned, the tumor immunity cell proximity mechanism, we bring the T cell into the tumor. And in fact, we have two approvals of this mechanism in solid tumors, tarlatamab for small cell lung cancer and tebentafusp for uveal melanoma. But we can actually do also a dual inhibition of different checkpoints, such as the ones you can see in the slides that are being studied. Or we can target tumor microenvironment mechanisms, resistance mechanisms, such as ivonescimab with the PD-L1 VEGF, or also targeting TGF-beta. There are multiple targets that are being studied both in early clinical trials and in late clinical trials, and this in multiple tumor types. The third modality would be cancer vaccines. There are different types of cancer vaccines depending on the antigens being anonymous or predefined, and if the stimulation is done in situ or ex vivo. But just to mention one of the successes, the Keynote 942, this is an individualized neoantigen therapy vaccine compared with pembrolizumab versus pembrolizumab alone in patients with resected melanoma. And you can see that patients that received the vaccine have really a recurrence-free survival that is much higher, and a distant metastasis-free survival that is much higher than the patients that received the pembrolizumab alone. So we'll also need to follow up with bigger results on this trial. But just to summarize, we do have different antigens and two different types. One, the tumor-associated antigens. So these are expressed in normal tissues, but overexpressed in cancers, such as cancer testis antigens, oncofetal antigens, viral antigens, and overexpressed antigens. And on the other side, we will have the tumor-specific antigens. So these are produced exclusively by tumor cells and most often by somatic mutations. And this can be private neoantigens, such as the one of the vaccine that we've just done, but also shared neoantigens, such as KRAS. And finally, we can also work on improving the tumor microenvironment in order to improve checkpoint inhibition blockade, because now we know that the myelosuppressive and the tumor microenvironment is really key on how checkpoint blockade works. So this is a very complex relationship, and a lot of targets are being studied. I just want to highlight CCR8 because I really think it's a very attractive target that is being studied right now. So CCR8 is highly expressed in the surface of tumor infiltrating Tregs, and the main role is really to participate in the recruitment of Tregs and Th2 cells in the tumor microenvironment. And there are multiple CCR8 inhibitors that are being studied right now in phase 1 clinical trials. Just to highlight this one that presented the results in ASCO LM108, and this was studied in patients with gastric tumors. You can see an overall response rate of 36%, but I do want to highlight that these treatments also come with some toxicities that we need to learn how to manage, mainly GI toxicities and AST and ALT elevations. Finally, just to mention that checkpoints are also being used as targets for other modalities of treatments such as B7H3. So B7H3 is really now being used as a target for monoclonal antibodies, but also for bispecific CAR T cells, but also for ADCs, and multiple ADCs being studied against B7H3. DS7300, it's probably the one that is more advanced. You can see here that the results that were presented in ESMO with an overall response rate of 32% in different solid tumors, and particularly high activity in patients with small cell lung cancer. And we also are studying B7H4 as another target. So a lot to be looking for. And with this, I really want to thank you for your attention, and I want to encourage to continue listening to the next chapter of Shaping the Future of Tumor Agnostic Strategy. Thank you very much.
Hello, my name is Atish Chaudhary, and I'm Senior Physician at the Dana-Farber Cancer Institute and Assistant Professor of Medicine at Harvard Medical School in Boston, Massachusetts, USA. In this chapter, I'm going to be talking about the future use of tumor-agnostic therapies in precision oncology. So to really understand precision oncology, we need to understand the biomarkers that we might look at that would suggest vulnerability for a particular treatment type. So those biomarkers can be DNA, RNA, IHC, or imaging. And so in DNA, you can look for mutations or translocations, copy number alterations, or genomic scars. In RNA, you can look for translocation or gene expression signatures. By IHC, you can look for a target, the overexpression or loss conferring vulnerability, and functional readouts. And then there can be imaging that's used to assess for target expression, signaling activity, and metabolic activity. So one example in using these different assays to assess a vulnerability is for homologous recombination repair deficiency. So this is classically assessed by assessing for a mutation in a gene involved in homologous recombination repair, but this can also be assessed in DNA through looking for chromosomal aberrations or mutational signatures. You can also look for the phenotype using gene expression for a BRCA1 or BRCA2 loss classifier, and you can also do functional experiments to look for focus formation of RAD51, which is involved in homologous recombination repair. So reviewing the tumor-agnostic drugs and targets that have already been approved, these include the TRK inhibitors, larotrectinib, entrectinib, and repotrectinib, the PD-1 inhibitors, pembrolizumab and dostarlimab, the BRAF inhibitor, dabrafenib, with the MEK inhibitor, trametinib, for BRAF V600E mutations, selpercatinib for RET gene fusions, and trastuzumab deruxtecan for HER2 overexpression by IHC. So in terms of looking for new targets for precision oncology, this can be based on inhibition of oncogenic signaling through direct inhibition of the relevant signaling molecule. So this includes mutated molecules like RAS isoforms FGFR, BRAF, and PIK3CA, fusions like NRG1, FGFR1, BRAF, and ALK, and also a variety of targets that can be overexpressed that leads to overactivity. In addition, you can target synthetic lethality. So there's classic synthetic lethality, and that can be divided into DNA damage repair based or non-DNA damage repair based classic synthetic lethality. And there can be something called contextual synthetic lethality, where particular gene expression changes or mutations might confer some vulnerability to metabolic inhibitors, for example. So a variety of DNA damage repair pathway inhibitors are being tested in clinical trials, as listed in the first column. And then there's a variety of epigenetic targets like EZH2, CDK9, and BET, and P300. And when there are particular epigenetic gene losses like SMARCA4, the cancer becomes dependent on its paralog SMARCA2, and that can be a target as well. Interestingly, particular mutations and gene losses can confer synthetic lethality a vulnerability to particular inhibitors. For example, MTAP loss can lead to vulnerability to inhibitors of PRMT5 and MAT2A. PPM1D mutations can lead to vulnerability to NAMPT, and VHL mutations to GLUT1. And then there are a variety of surface targets that can be targeted by a variety of strategies, for example, through antibody drug conjugates, radiopharmaceuticals, bispecific T cell engagers, as well as CAR T cells. That's been discussed in the immune therapy approaches before this. And many of these targets are expressed across a variety of cancer types. And so these are very interesting and novel targets to develop tumor agnostic strategies in the future. So in terms of specific emerging tumor agnostic therapies that are currently in trials, these include drugs that target KRAS G12C, NRG1 fusions, TP53Y220C, FGFR, ALK, and then drugs that target ALK, ROS1, and Anthrax 123, RET, and TROP2. So these are all being tested in tumor agnostic indications. But there are several challenges in tumor agnostic drug development. Some of these are biological. So some of the biological challenges include the biomarker. So is this something that is homogeneous across the tumor burden, or is there subclonality or heterogeneity? And then there can also be tumor-specific cut points for continuous biomarkers. So tumor mutational burden, IHC scores, AHRD scores actually might have different cut points across different cancer types. There can also be issues around drug delivery and the tumor microenvironment. Some cancer types are more vascular than others. Some cancer types are associated with some stromal barriers. There can be drug breakdown or efflux in particular tumors, and there can be hot versus cold tumor microenvironment for immune therapies. And then resistance mechanisms can also be different across different tumor types, for example, related to co-occurring mutations or adaptation to prior treatments. And there can be also upregulation of compensatory pathways, like upregulation of compensatory EGFR signaling in BRAF V600E mutated colorectal cancer that leads to relative inactivity of these agents in that particular cancer type. But there are also statistical and regulatory challenges as well. So, for example, these trials require a presupposition that biomarker-based classification is more fundamental than organ site-based classification. And so you generally want a robust and irreversible biomarker for conferring drug sensitivity and pretty robust preclinical and clinical evidence for activity that's based on that biomarker. You'd require a prospective statistical design to allow selection of multiple indications. And so the Bayesian hierarchical model is commonly used along with various variations and extensions. And it also requires a unified framework of tumor agnostic potential. So for example, ESMO established this tumor agnostic classifier and screener, or ETAC-S, which is some minimal eligibility requirements for a tumor agnostic potential. So first, you need robust preclinical evidence of mechanistic and biological rationale, and then you want phase 1/2 or phase 2 trials where 2/3 of the tumors investigated and greater than or equal to 4 tumor types have a minimum overall response rate of greater than or equal to 20% and greater than or equal to 5 evaluable patients with refractory disease as a minimum requirement for the establishing the potential. But there are other frameworks as well. So in terms of other challenges, these trials are generally single-arm, non-randomized phase 2 trials. So you can't validate the biomarker if no biomarker negative patients are enrolled. This leads to approval when no other treatment options exist, and you actually require new trials for earlier lines of treatment or in the neoadjuvant or adjuvant setting. These trials have small sample sizes for individual tumor types, so there's a lot of uncertainty regarding benefit in tumor types that are not well represented in the studies. And then there can be post-marketing requirements like increasing the sample size for certain tumor types, looking for longer durations of response, additional data on the optimal dose, specific populations, and companion diagnostics. But despite these challenges, it's a very exciting time in precision oncology, and we're very much looking forward to where the future leads us. Thank you so much for your attention. And please continue to listen on to the next chapter on future directions in precision medicine for non-small cell lung cancer.
Good afternoon. My name is Jurishka Naidu, and I'm a professor of medical oncology and an adjunct professor at Johns Hopkins University, as well as a consultant medical oncologist at the Beaumont RCSI Cancer Centre in Dublin in Ireland. And today I'll be presenting on future directions in precision medicine for non-small cell lung cancer. And one of the core concepts is that molecular testing identifies the genomic landscape of lung cancer. We see that over the last 10 years, historically, patients with lung cancer were assessed simply by immunohistochemistry and subtyped into their morphological appearances of lung cancer, adenocarcinoma, squamous carcinoma, and others. But in recent years, it's become a standard of care to do next-generation sequencing on all of these patients. And we see here an ever-growing list of molecular targets that can lead a patient to a more precise form of treatment in which there is greater efficacy for genomic subsets of lung cancer. These alterations, as mentioned before, therefore lead to a range of targeted therapies, and even within the specific types of targets, there are several different options and several different modalities to which targeted therapies can be applied in that setting. Good examples include EGFR mutant lung cancer and ALK rearranged lung cancer, and an ever-expanding list of these targets. One of the core concepts around targeted therapy, however, is this universal mechanism by which a targeted therapy may lose its efficacy in patients with oncogene-addicted cancers, the stage of acquired resistance. And we see here in this paradigm of how we think about oncogene-addicted cancer that there are specific clones or a dominant clone through which the targeted therapy is most effective. When a targeted therapy is given, most of these clones may respond to treatment. However, at a certain time point, they acquire resistance to therapy by a number of mechanisms, but sometimes through the development of quintessential bypass pathways. We can also see that certain oncogene-addicted types of cancers, and lung cancer is a good example, may be immunotherapy responsive, some may be immunotherapy unresponsive, and some may be both, depending on the range of genomic features that they may harbor. So there is a standard thought process that oncogene-addicted cancers respond to targeted therapy and immune-related or immunogenic cancers respond to immunotherapy. But in truth, there is an overlap between these subsets. And we see in lung cancer, for example, KRAS mutant lung cancer can respond to immunotherapy depending on the presence of co-alterations, and some subsets may not. Similarly, BRAF is a good example of this, where it can be responsive to immunotherapy or not, while EGFR mutant lung cancer and ALK rearranged lung cancer that traditionally does respond to targeted therapies may not respond to immunotherapy. We see here that there is a range of emerging targets with respect to non-small cell lung cancer, and this is an example of the complex numbers of pathways that may be implicated in which cancers may respond to which targeted therapies. And as you can see, this is an ever-growing list of targeted therapies that we need to keep on top of. A very good example of this is HER2. HER2 is a molecular alteration seen in a small subset of patients with lung cancer. And what's very important to understand is that HER2 alterations in the context of lung cancer come in different flavors. We can get HER2 point mutations, we can get overexpression of HER2 through IHC, and then we can get a range of other alterations such as HER2 exon 20 insertions. And there are a range of targeted therapies that may be suitable for these different subsets of HER2 activating mutations or alterations. In the subset of patients with HER2 activating mutations, which accounts for about 1% of non-small cell lung cancer, a good example of a new targeted therapy that is emerging is the BAY2927088 molecule. This is an orally bioavailable mutant-selective small molecule dual kinase inhibitor. And we see here that it has selective activity against HER2 activating mutations over wild-type EGFR. This is an example of a phase 2 study of this agent, the SOHO-01 study. Another example of an EGFR-specific tyrosine kinase inhibitor is zongertinib. This selectively and covalently bonds to the tyrosine kinase domain of both wild-type and mutated HER2 receptors. And we see here that this agent has now received FDA breakthrough designation for the subset of patients that have HER2 mutant non-small cell lung cancer. This is based on the BEAM-Lung-01 study. Another good example of advances in precision medicine in lung cancer is the subset of patients with BRAF mutant non-small cell lung cancer. What's interesting about this subset of lung cancers is that even within the BRAF mutation, there are various different subsets of mutation that should be treated differently. We see that class 1 mutations, the classic V600 mutations that we associate with melanoma, happen in about 40% of cases, and that about 50% of these are never smokers. We see that class 2 and class 3 BRAF mutant lung cancer behaves very differently and may have co-alterations in RAS and associated with more of a smoker's phenotype, associated with the development of brain metastasis and occurring more commonly in patients who are former or current smokers. We see, based on these different classes of BRAF mutations, that a range of different single agent and combination approaches are currently under study for BRAF mutant non-small cell lung cancer. Another area that I think is important to touch on is the area of RAS mutations. We know that RAS mutations are present across a number of different tumor types depicted in this figure, ranging from the most common, such as non-small cell lung cancer, pancreatic ductal adenocarcinoma, and colorectal cancer, but less commonly can occur in head and neck cancer, thyroid cancer, melanoma, and bladder cancer. Importantly, we see from this figure that the incidence of allele-specific subsets of KRAS mutant lung cancer differ by tumor type, with KRAS G12C being the most common in lung adenocarcinoma, but not as common, for example, in pancreatic ductal adenocarcinoma. Focusing specifically on lung cancer, we see that now it is important to not only understand the specific allele of KRAS mutation, so a G12C, G12V, or G12D, but now it also is important to understand the co-alterations. So we are now evolving to pie charts within pie charts in order to understand which specific subsets of patients may benefit from certain targeted therapies, while others may benefit from immunotherapy or immunotherapy-based combinations. KRAS targeting is also extending beyond KRAS G12C to other different allele-specific subsets. We are seeing specific targeted therapies being developed for KRAS G12D mutant lung cancer and KRAS G12V, as well as pan-KRAS inhibitors in this setting. So with this, it's a very exciting time to be in precision oncology for non-small cell lung cancer. I want to thank you for your attention to this chapter, and please continue to follow on to listen to the next chapter on future perspectives of liquid biopsy in precision oncology.
Good morning. Today we will have the opportunity to discuss together some aspects regarding future perspectives of liquid biopsy in precision oncology. My name is Umberto Malapelle. I'm the chair of Predictive Molecular Pathology Lab at the Department of Public Health from University Federico II of Naples. And so we can start to discuss together the application of liquid biopsy in solid tumors. As you can see in this slide, this is the first statement paper of International Society of Liquid Biopsy, in which we identify 3 areas of implementation of liquid biopsy in clinical practice, starting with lung cancer, colorectal cancer, and breast cancer. But today we have other area. Starting from non-small cell lung cancer, as you can see in this slide from the recommendation of the ISLC, we can use 3 different approaches. One is sequential one, the other one is complementary approach, and the third one is a blood-first approach. But we need to take into account the structure of our national regulation to implement one of these approaches. But on the overall, when we are able to integrate the results coming from liquid biopsy and tissue ones in non-small cell lung cancer, as you can see in these slides, in example in Europe, we are able to recover more than 6,000 patients each year for a target treatment in clinical practice, carrying a mutation in EGFR, KRAS, BRAF, ALK, ROS1, RET, N3K, and so on. So this is a really relevant part, but the final goal of liquid biopsy is not to have an aesthetic vision on the presence or the absence of a particular type of alteration, but is what we can see in this slide. So the possibility to follow up during the time the clonal evolution in blood, starting to analyze different type of alteration and to follow this type of evolution in conjunction with the tissue-based analysis and the imaging-based analysis that you can see in the right part of these slides. On the overall, by using liquid biopsy, we are able to design the path that you can see in this slide. Starting from the right side, in which we have the possibility to introduce liquid biopsy metastatic setting, but the idea is to move the liquid biopsy in treatment guiding, minimal residual disease, early detection, until to cancer interception. This is possible only in a picture, or we can really apply this evolution analysis in liquid biopsy in solid tumors patients. And as you can see in the slide, this is a patients with non-small cell lung cancer for which we don't have enough tissue in the starting point, and we decide to move in liquid biopsy. As you can see, we are able to follow up the evolution of mutant allele fraction during the time and the quality of the appearance of the mutation that you can see in the right part of these slides. This is not only for non-small cell lung cancer, but in this slide you can see the case of ECR1 mutation detection in breast cancer. And if we see all the mutation reported in the graph, only ECR1 has this type of difference between primary tumors and metastatic sites. So this means that after a line of treatment with an aromatase inhibitors, we can detect ECR1 mutation starting on ctDNA extracted by liquid biopsy. And another important part is that we need to test and retest our patients during the course, because the long exposure to aromatase inhibitors increase the rate of ECR1 mutation. And we can detect this one by using liquid biopsy. Pulling all together the topics that we have discussed, we are able to design this graph that you can see in the upper part, following the entire journey of a patient by using liquid biopsy. But we need to overcome some barriers that you can see in the lower part of these slides, in particular when we move in the early detection setting, in minimal residual disease, and in cancer interception, because we need to improve our resolution power, because the quantity of ctDNA, as we can see in the graph, is lesser when we move in the early stage. And so for this, we need to improve our technological ability to see not only mutation on ctDNA, but also other type of alteration, like as transcriptomics, so at mRNA level, methylation, so epigenomics, and then protein. So the idea is that liquid biopsy doesn't mean only ctDNA, as we can see in this cartoon, because we can complement the results coming from CTC, ctDNA, cell-free RNA, extravesical cargo, and so all the other parameters that you can see on the right part in these slides can be complemented to improve our resolution power, to give us the opportunity to move liquid biopsy not only in metastatic setting, but also in the other setting that we have discussed. And the final goal is to move beyond too early detection in the cancer interception. So this means that the dream is to use liquid biopsy in the overall population to detect this type of alteration, to complement the results, and to prevent the evolution of a particular type of tumors. So thank you very much for the attention, and please continue on to answer the questions that follow and complete the evaluation forms. Thank you very much.
This program was presented by Medscape Education Global.